So yes, in the last portion we discussed about the cyclic and non-cyclic phosphorylation, and this is the Z scheme, and Z scheme is the non-cyclic photophosphorylation. Okay, so this is the P680 means photosystem 2, P700 means photosystem 1. So you need to understand that. Now, so if I talk about the ATP synthesis during the light reaction, of course we know that. So in the non-cyclic phosphorylation, ATP and NADPH both are getting produced, and in the non-cyclic phosphorylation, non-cyclic, only ATP is getting produced, right? Okay, so actually I will explain you something. Actually, this is known as the chemio-osmotic hypothesis. So before okay, firstly just see, chemio-osmotic hypothesis is just the I just explain you the how the ATP is getting formed. Okay, so actually chemio-osmosis requires a membrane, a proton pump, and a proton gradient and the ATPase. See, Vishnu, there is a phenomena which is the chemiosmosis, and this chemiosmosis help in the production of ATP. So it need a membrane, a proton pump, a proton gradient, and the ATP. And what is that? I will explain you. See, can you just see this membrane? See, this is the ATP, Vishnu. This is the ATPase and proton pump. Can you just see inside there are so many hydrogens, H plus ion.
Yeah, but so
So,
cation, so
so
plus
yeah,
proton.
yes. Inside the membrane, there are so many H plus, and outside there will be very less H plus. So there is a proton gradient between them. Yes or no? There is a difference.
Yeah.
So we obtain the one thing. We have the membrane. This is the
What
membrane of thylakoid.
significance does the gradients make?
I will tell you. I will tell you. I will tell you. This is the membrane. This is second use. Because of the proton pump, we have just put the so many H plus inside to make the proton gradient. Okay, now what is the use? Okay, can you just see this molecule? This is written over here, F naught and F1. This F naught and F1 molecule is also known as the ATPase. It is an enzyme which help in the production of ATP. And how? I'm telling you, whenever whenever two H plus ions are passing through this molecule, whenever two H plus are passing from this F naught and F1 molecule, so they will help in the production of one ATP. It is the rule for the ATPase. Rule for the ATPase.
Can you say it
So
again? What
yeah,
what did
yeah,
you
yeah. I'm
say?
telling you, I'm telling you. Inside the membrane, we have created so many H plus ions, and outside there is very less because of the because of the proton pump we have done that. Proton pump has given the so many H plus inside the membrane. Okay, now proton gradient is created. Proton gradient means proton difference. Inside edge a more H plus, outside less H plus. Now, so they want to come outside, right? To make it balance, they want to come outside, so they have to come outside only through this pathway. Can you just see that? This is the F naught F1 molecule. This is called the ATPase. So whenever 2H plus will come outside, whenever 2H plus will come outside, so they will form ADP to ATP. Okay, so it means when the 2H plus is coming outside from this gateway, so it will produce the ATP. This is how they are helping the production of ATP. So
So let's
remember,
say their H pluses want to come back in. How will they come in?
in because of the proton pump.
Yeah, but
Proton,
like, do they go into the same place that they come out?
no. There is like here there will be a proton pump and they are coming. Can you just see that? There is a arrow,
Oh,
cytochrome B.
that's where it comes from. Okay, I see.
Yes. So there is a complex,
Oh.
say cytochrome B and F. So because of this
Mhm.
pump, the H plus will be taken inside. You remember when the water is getting photolyzed, 2H2O, it is forming 4H plus something something, and then half O2 and then electron. Electron is going into the ETC, electron transport chain. Oxygen is coming outside because we say in the photosynthesis oxygen is released. It means oxygen is coming. But what happened to the hydrogen? Hydrogen is coming inside in the chemiosmotic hypothesis. Okay, and they are producing the ATP for the dark phase because glucose will be formed. Getting something something. Okay.
Yeah, I'm getting
So,
it.
remember, yes, this is the chemiosmotic hypothesis. So you need to remember. And I told you ATPase contain the two partner, F naught and F1. So F naught is embedded in the membrane. See, F naught, F naught is embedded in the membrane, F naught, and F1 is coming little bit outside the membrane. This pink is a membrane.
Yeah, I'm
So So this F0 and F1, it is called the ATPase. And why it is called ATPase? If I ask you, Vishnu, why it is called ATPase?
because it has two parts.
No, because they
Oh
help
no
in
no no
the.
no. Okay, no. Because it's coming out. The H plus is coming out, right?
But why it is called ATPase only? Why not H plus A's? Because
Uh
they help in the production of ATP.
oh.
When the two hydrogen are coming, so it is
Yeah, I
producing
know. I
ATP.
said like when they're coming out, like when the H plus are coming out.
Okay, fine.
Mm.
So they are called the ATPase because they help in the production of ATP. Simple. So the breakdown of proton gradient, proton gradient means when the 2H plus ion are coming outside, it means proton gradient is going to break. So they will produce the ATP. So this ATPase means F naught and F1 molecules. So they help in the production of ATP. Fine.
Yeah, the ATP ace is formed when the H's in the middle right there come out, and then they form the ATP synthase, also known as
No, 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 no,
ATPase.
no, no, no. Not formed ATP synthesis. ATP synthesis is already there.
Oh no,
Already,
okay.
already
They
there inside the
it
membrane.
comes
And
out.
yes, whenever 2H plus comes out, then it
Yeah,
form the ATP. Okay, it is permanently present there, F naught and F1, which is called ATP synthetase. Okay, F naught
it's
and
just when it
F1.
comes out.
Yes. So when the 2H plus comes out from the F1 and F naught molecule, F1 and F naught molecule is also called ATP synthetase. So it formed the ATP. Okay. So firstly, we are intentionally making the difference between the H plus, and then when it is coming outside to make it balance, then it formed the ATP. Okay.
So does one oh, wait.
Yeah,
Oh,
yeah.
okay, I see.
Any doubt?
I mean, which hole does it come out of F0 or F1?
F naught and F1 is a completely one molecule which is combined together.
Okay.
It is combined together. It is coming out from the F naught and then F1 H plus,
Oh,
and then
yeah,
it
okay, I
formed
see.
the
I'm not good at reading diagrams.
adenosine diphosphate to ATP. Okay, so it is
It looks
a
like a penguin to me. So,
okay. So it is a completely one molecule which are going to attach together, something like this. So whenever H plus is coming outside, so they have to pass from both the molecule, and this
okay.
combinedly known as ATP synthetase. It is the another name of F1 and F naught. Okay.
Oh, so the molecule F0 and F1 is called ATP synthase.
Yes.
Oh,
F naught, F1 together form a molecule which help in the production of ATP. So that's why it is also called ATP synthetase. Okay, got it now? I can't hear your voice actually.
yes, I was writing it down.
Okay. So, yes, I already told you about all these, like photosynthesis take place in the two-step light reaction and the dark reaction. I told you already this thing. So, light reaction is a light-dependent reaction. It occurs in the thylakoid. I showed you thylakoid because the energy production was there, na? So that was the thylakoid because the H plus are coming and the ATP is getting formed. I told you, and it result in the light reaction is the production of the assimilatory power NADPH and the ATP and oxygen. I told you this thing from the water H plus that is helpful for making the gradient. O2 is coming, which is good for us, and electron is coming. It is producing the ATP and NADPH, right? If it is a non-cyclic phosphorylation, it
Yeah.
is producing NADPH. So all these things are happening in the light reaction. But now we are coming to the dark reaction, right? So it is dependent on the product of light reaction because
Yeah.
without all these energy, dark reaction won't happen. So dark reaction is depending on the light reaction, but we will not say depending, we will say product of the light reaction.
I
It
could
occur.
pretend photosynthesis is like bodybuilding, light
Yes,
reactions eating and dark reactions working out.
yes, exactly. And it take place in the stroma. Stroma means outside the thylakoid membrane liquid.
Mm-hmm. Yeah.
And it is also called the biosynthetic phase, remember. Dark
Okay.
reaction is also called the biosynthetic phase. So of course here, sugar means carbohydrate. Don't say that sugar, it is just the carbohydrate means glucose, so it help in the production of glucose, which is a sugar. Okay, find
Mm-hmm.
that.
Mm-hmm. Mm-hmm.
So now we are going to study light reaction or dark reaction.
What did you say?
Now we are going to study light reaction or the dark reaction,
Hopefully both.
because we have studied the light reaction na.
I,
So
I know.
we are.
Hopefully we can study even more of both.
Okay, 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 got it. Now we are coming to the dark reaction, because we have studied about the how the production of energy and how the photophosphorylation take place. So now we are coming to
Now
the.
we're going to look at how the glucose forms.
Okay, yes. So this is the phase. It is also known as the biosynthetic phase. I told you recently just. So
Yep.
dark phase is also called the biosynthetic, means synthesis of something, and synthesis of something means glucose. So here carbon dioxide is reduced to the carbohydrate. You know it. CO2 is converted into the C6H12O6.
Yes, sir.
So photosynthesis in this phase, carbon dioxide is assimilated, or better to say reduction. Reduction means adding of hydrogen. So
Mm-hmm.
yes, it will form the C6H12O6, and it take place at which site? Remember, stroma. And that light reaction take place at which site? Thylakoid. Okay,
Okay.
thylakoid, that coin like structure I told you.
The
So
coins, yeah,
light
stack of
reaction
coin.
take place in the thylakoid, but the dark reaction take place in the stroma. So
Oh, they're very close to each other.
yes, almost very close. Like one is thylakoid and outside is stroma. Yes.
I see. Okay. So dark reaction stroma,
Yes,
light reaction thylakoids. Okay.
take place in the thylakoid. Yes.
I see. Okay.
Now we have the two pathways. Okay. See, we have the in the plants we have the two kind of pathway by which they form the glucose. First is the C3 pathway, and other is the C4 pathway. Carbon 3 and carbon 4 pathway. So we can form the glucose by the two modes. Okay, so first is the Kelvin Benson cycle, which is also known as C3 cycle, and the second cycle was given by Hatch and Slack pathway, which is also called C4 pathway. So in the dark reaction, we have basically the two things, C3 and C4. So C3 is also known as Kelvin Benson cycle, and C4 is Hatch and Slack pathway. Okay, okay, Vishnu.
Yeah.
So now we are going to study about the first Kelvin cycle, which is the Kelvin Benson cycle, and it is a C3 cycle. Why it is called C3? Because in this cycle, the first product is always the C3. I mean carbon number 3. It contains the only 3 carbon compound, the first product. That's why it is called Kelvin cycle. So Kelvin cycle is found in all the photosynthetic plants. In all the photosynthetic plant, Kelvin cycle. And in this pathway, the source of the carbon is carbon dioxide. It means whatever the compound they are going to form, the source of carbon is carbon dioxide that enter into the cycle, and it is getting reduced and it is forming the carbohydrate. So it means in the Kelvin cycle carbon dioxide is entering, and when this carbon dioxide is, because H2O we have already seen that. Okay, from the H2O, O is producing O2 to us, electron is helpful for production of energy, so H plus. So all those H plus are combining with this and they are forming the carbohydrate, remember, because in the photosynthesis water and carbon dioxide both are required, and they will form the glucose. So they are coming and they are forming the carbohydrate. So carbon dioxide providing the carbon. Very simple it is, right? So remember that these things is just the history that you don't need to remember. But yes, this one. So C3 cycle have basically the three main processes. First carboxylation, second reduction, and then again coming back, which is called regeneration. So C3 cycle have the three main step. Carboxylation is indicating you like carbon dioxide is getting added. Reduction means, it means in this step glucose is getting formed. Reduction, because when the carbon dioxide is reduced, so it form the glucose. And regeneration means something is there so that cycle can remain continue. Regeneration means whatever it was there, whatever the reactant we had, so we are going to form it again. This is called the regeneration, so that cycle keeps on continue. So why is Kelvin cycle is called C3 cycle? I told you about that.
Because there's 3 steps.
Because the first product is
Oh,
of 3
C3.
carbon.
Yeah.
Okay, so first is of the 3 carbon, that's why it is called C3 cycle. See, first
Okay, I don't
listen.
understand this
Okay, okay,
diagram.
I'm telling you. Firstly, we have to start it from here. This is called the RUBP, ribulose bisphosphate. This is the most common, you can say, reactant in the plant. So it is the RUBP. RUBP, it means ribulose bisphosphate. One second. Can
Mm,
you hear my voice?
right. Yeah.
Okay, okay. So ribulose bisphosphate we have. So carbon dioxide is getting added into that. So carbon dioxide is adding, that's why it is called carboxylation. I told you
Mm-hmm.
carbon dioxide is coming. And this RUBP, it is of 5 carbon.
Mm-hmm.
So 5 carbon and one more carbon. So this is the carboxylation. So this 3 PGA is getting formed. 3 PGA. And it is of only 3 carbon. It is of only
Yeah.
3 carbon. So just tell me how many molecule are getting formed? It is 5. One is here. Total 6 carbon were there. And now it is only formed the 3 carbon. So it means how many molecule of 3 PGA will form?
3 divided by 2 is 6. Or I
Because
mean,
yes,
wait,
one
what? That didn't make
one
any sense.
one PGA contain 3 carbon and we had 6 carbon. So how many molecule will get formed? Only 2 na?
2.
3 and 3. So it means
6
from,
divided by 3 is 2? Math? I don't know.
yes.
I haven't taken math
Okay.
in like a year.
Okay. So this is the two molecules of 3 PGA will get formed. Okay. This is the carboxylation. And now from the 3 PGA, you are doing the reduction of that. And now it is forming triose phosphate. Remember, I will tell you what
Oh.
is happening. Just you have to just remember the cycle now. So from the reduction, it is forming triose phosphate. From the triose phosphate, it is giving one carbon to, see, this triose phosphate. It is giving one carbon to the downward side, and now see, total 6 carbon were there, and now it is forming triose phosphate, two molecule. So finally, from the triose phosphate, one carbon is going down. So now, how many carbon total over there? How many total carbon?
Wait, say it again?
3 PGA,
Okay.
two molecules of
Up to that
3
pink there's 2 right there.
3 PGA contain total 6 carbon. क्योंकि two molecule, because two molecules were formed. So
Yeah.
3 and 3, total 6 carbon. And
Yeah.
now when they are going to reduce, so after the reduction, one carbon is coming down. So one
Oh,
carbon
so
is coming
4?
down. So total how many carbon were formed?
4?
Only one carbon is going down from the one molecule only. Only one molecule, not the other one. So total how many carbon were there?
5?
Yes, five. Five. So these are going to
Okay.
combine. These are going to combine, and again regeneration. They are forming
Wait.
RUBP. Wait a second. So RUBP contain the 5 carbon, yes or no?
Yeah.
And so one is going down. So this is the what, this is how this cycle is continue, regeneration. So just tell me, glucose is actually made up of 6 carbon,
Mm-hmm.
and in the one cycle you just given down the only one carbon.
Yeah.
Glucose is getting formed downward, here
Yeah.
glucose will be formed downward. So how many cycle it has to run for giving one
6.
glucose? Yes, very good. Because one cycle is giving only one carbon, so second cycle will give one carbon again. So 6 times it will give you this 6 carbon, right?
Oh wow.
Okay.
So cool.
So I was just explaining about that. The first step was the carboxylation. So the first compound which is formed, I know it is a two molecules, but if I talk about the one molecule, so one molecule contain how many carbon?
Two.
What?
Oh, three. I thought you were pointing to the number of like total. That pink
Total
one?
total molecule of PGA is 2. But
Yeah.
one molecule contain how many
Three.
number carbon? It is a 3.
Three.
That's why it is called the C3 cycle. Because the first product contain the 3 carbon, that's why the cycle name become the C3 cycle. And this 3 phosphoglycerate is also known as PGA. PGA.
Yes.
Phosphoglyceric acid it is also known as. So you can say PGA. Okay. Okay, so for the formation of glucose, this cycle has to run how many times?
Six.
6 times. And so one cycle is giving you the how many carbon for the
One.
glucose? One. Okay, very good. And this reaction is taking place in how many steps? Carboxylation, reduction, and
Three.
regeneration. Yes, very good. Well done. Okay, now can you just see that? Can you just see that? It is producing the energy or it is taking the energy?
What'd you say?
It is producing the energy or it is utilizing the energy.
Producing?
No, because dark reaction only for the production of glucose.
So it's
So
using up the
yeah,
glucose.
yeah, it is using the energy which
Okay.
were formed in the light phase. Remember,
Yeah, the thing where the proton pump came out.
photophosphorylation. Yes,
Yeah,
photophosphorylation. Okay,
that one.
zigzag one and the circular. Okay,
Yeah. I remember. I just forget the
so can you
names.
just see ATP and
Yeah.
NADP? They are getting used up and they are forming NADP and NADP plus. So it means they are
Hmm.
using the energy.
What's
Whenever
PI?
PI means phosphate.
Oh,
From
I
the
see.
triphosphate, it is forming diphosphate and phosphate. So it means they have utilized the energy. Whenever
Mm-hmm.
ATP is converted to ADP, it means they are utilizing the energy. Okay, and
Mm-hmm.
whenever ADP and phosphate is combining forming the ATP, it means it is the production of ATP. Okay, okay.
Yeah.
So ATP and NADPH, they are both uh like so it means this cycle is completely utilizing. So both the energy currencies are utilizing by this Kelvin cycle and they are helpful in forming the ADP and NADP. Got it, everybody? Okay. So in the
Yeah.
regeneration, again, they are utilizing energy or they are producing energy.
Oh, they're utilizing it.
Yes, they are again utilizing. So it means that statement was completely right. What? Ki biosynthetic phase, dark phase, dependent on the product of the light phase because the product of light phase is energy, ATP.
Mm-hmm.
Okay. So Vishnu, are you getting what I'm saying?
Yes.
Okay. See this line. Dark phase is saying that it is dependent on the product of light reaction. And in the light reaction, NADP and ATP was produced. So it means dark reaction is dependent on these product, dark reaction.
Yes.
And we,
Dark reaction is dependent on the light reaction.
yes, a light reaction product. Yes, you can say light reaction
Yes.
also, but
Yes.
mainly the product. And yes, so because without energy, it will never happen. So this energy is coming from the light reaction or dark reaction, this energy or NADPH.
Or light reaction.
Very good. Yes.
Yay.
Good. So this is the Kelvin cycle. Okay. So finally you can see that. Okay. So for the one glucose, I told you already for the formation of one glucose, how many CO2 will be needed? Can you just see that this CO2?
Yeah, you need six.
Yes, because you are running the cycle 6 times. So of course you need six molecule of CO2. That's why in the reaction, you always write the six CO2. Remember?
Oh yeah. Well, now I understand it.
Yeah. So ATP required. So for fixing one molecule of CO2, so you need three, I mean two for reduction and one for the regeneration. For one molecule of CO2, fixing one
Wait. So
molecule of CO2.
NADPH is the H is the nicotine and oh my god, I forget the name.
Okay,
Is
okay,
that the
okay.
one that's worth 3 ATP?
Wait a second, wait a second. I'm just telling you. ADP. See this cycle. See very carefully. One ATP is for the one cycle. For the one cycle, one ATP is getting produced. I mean, one ATP is getting utilized, and here. So it means in the one cycle, how many ATP is getting utilized?
A one. Two, 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 two.
Two. One is ATP here, and other ATP is here. So two. So
Yeah.
it means for this six cycle, how many ATP will be utilized?
12?
Yes, 12. So it means 12 ATP is getting utilized. One second. Why are they writing like for fixing 6 molecule of CO2? 3? Why are they writing 3? One second, one second. I guess we are doing some mistake.
Maybe because there's 3 steps.
Oh my God. Yes, 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 yes. I'm telling you, I'm
Yeah,
telling
there's
you.
two. Okay, right.
Yes, because this is basically for the one na, but here it is a two molecule. Two molecule. So
Yeah.
see, for the carboxyl, I told you, two molecule of PGA is getting formed, two molecule, and then here. So it means if one molecule is coming here, so it is forming it is taking one ATP. So they actually here two molecules of PGA were there. So for the two molecules, so it would be two ATP and two NADP. But here they are going to combine. So now you don't need to take two here because they have already combined together. Okay. So here you have to
Mm-hmm.
write 2 and 2 because it is two molecule, 2 2. Okay. So it means 2 ATP is getting formed and 2 NADP is getting formed here. 2 NADPH is utilizing here. So just tell me total how many ATP utilize in a one single reaction?
Six. Or three, three, three,
3.
three.
2 ATP here and 1 ATP here because it is already combined na. It is not
Mm-hmm.
2. It is combined because one is giving down and they are going to combine. That's why it is becoming a 5 molecule single structure. So 2 ATP here and 1 ATP here. So 3 ATP in a single cycle. So for this six cycle, how many it will be?
18.
Yes, 18. Na? See, it is written over here also. 2 for the reduction and 1 for the regeneration. Isn't it? 2,
Mm-hmm.
2 for the reduction and 1 for the regeneration. Okay, got it now? Okay. So for the six molecule of CO2, of course, it will be 18. Na? In one cycle, 3 ATP is there. So for the sixth cycle, it would be 18 ATP. Am I right?
Mm-hmm.
Now, just tell me, can you see NADP? So it is the two molecule. So of course, NADPH is also two molecule. But can you see anywhere the same molecule? No. So in a one cycle, how many NADPH is getting produced? Getting utilized?
Just one? Or two, 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 two.
Two, two. Here it is
Because
two,
there's like
right?
two. Yeah, two,
Yes, two
two.
molecules are there. So, so if one cycle is taking two NADPH, so it means six cycle will take how many NADP? 12. 12 NADPH, right?
Mm-hmm.
So it means the complete net reaction. Can we say that? Because, okay, so six time you need
Yeah.
six RUBP. For the one cycle, you need one RUBP. So for the six cycle, you need six RUBP. So that's why it is written over here. Six RUBP, six CO2, 18 ATP, 18, 12 NADPH. So now they are forming the glucose. Glucose. ATP is forming the ADP and phosphate.
Mm-hmm.
Triphosphate, diphosphate, and single phosphate. So it is 18 ATP is going to broken down into ADP and phosphate. NADPH is going to broken down into NADP plus. 6 RUBP
Mm-hmm.
remain the 6 RUBP. So this is the complete cycle of photosynthesis. Are you getting that, Vishnu?
Yes.
This is the C3 cycle. If I ask you the one thing, why Kelvin cycle is also called C3 cycle? Tell me.
Why? Okay. Because in the initial reaction, see, there's three molecules of carbon released in the initial stage.
Yes, because the first product is of 3 carbon. Okay.
First product is three carbon.
Yes, carboxylation is the first step, na? So just after the first step, which is the carboxylation, it is forming the first compound. It is made up of three carbon. It means 3 PGA, 3 phosphoglycerate is made up of three carbon only. That's why
Hmm.
it is C3. Okay, and in the one cycle, how many ATPs are used? In one cycle,
In one cycle, three.
three, because two at which side?
Two.
Carboxylation, reduction, or
Reduction.
regeneration? Reduction. And one at,
Regeneration.
okay, after the regeneration, which product you will get?
ADP.
No.
NADP.
After the regeneration, which you will get? Which compound you will get?
What?
RUBP after the
Oh yeah, yeah, yeah.
regeneration.
That. Yeah.
Okay, after the regeneration, you will get RUBP na. This is of five carbon, and one
Mm-hmm.
carbon dioxide is coming, so total becomes six, and six will be divided equally.
And
Two
it's a loop
molecule.
again. Yeah.
Yeah, so it will remain continue. Okay, okay. Now you have to see how does high intensity affect the rate of the photosynthesis. See,
It's a
whenever the
concentration of light.
yes, see. If
Okay.
intensity is good enough, then of course photosynthesis rate is increasing. But
Oh, it's like the
if it
red blue. No.
if intensity become very high, intensity very high, then chlorophyll may be deteriorated. You can say
Okay, I
chloro
see.
it is a like chlorophyll will not work. See,
Mm-hmm.
in the winter, taking heat air is good. Heater air is good, but extreme heat air is good or not? No.
No,
Extreme,
too much is too bad.
yes, too much. It can burn your skin. So same thing is happening here. So good intensity is good, but very high light intensity, it is not good for the photo. I mean, not good for the chlorophyll. So if chlorophyll is not working now, chlorophyll is completely burnt. Okay, now chlorophyll is not working because of the high intensity. It is not functioning properly. So photosynthesis will happen. No.
No, it's
So
too
it
like
means
intense.
after the saturation, after a limit, after the saturation, it is not good. Can we say that?
Yeah.
So up to a limit, it is good, but after the limit, it is not good. Okay. So now we are coming to the next cycle, which is called the C4 cycle. But before the C4 cycle, I want to tell you, see, plants, like there is another phenomena with
Hmm.
C3 cycle we studied. Now we are coming to the C4.
Is this the neurovascular bundle or
Yes,
something?
it is a special type of cell arrangement in the certain plant, which are undifferentiated mesophyll cell surrounded by the large size bundle sheath cell, and it is good for the plant. Okay, so it is a very good arrangement. Can you just see that? Bundle sheath cells are there, and it is good. It is very helpful for taking the light, and it is very helpful for the production of glucose. So whenever any plant has the Karan's anatomy, so it means it is very good for the photosynthesis. So it is a special type of cell arrangement in which the cells or in which the certain plants like they have the mesophyll cell that is surrounded by the large size bundle sheath cell. So can you just see that large size bundle sheath cell, these? So when they are surrounding by the large size bundle sheath cell, so it is very good for the photosynthesis. So having Karan's anatomy, it is good or bad for the plant?
Is it good?
Yes, it's good. So when the
Wait, is
mesophyll,
that? Oh my
see, see,
god! It
see,
looks like a
in
wreath.
the C3 plant, mesophyll cells were there, but a mesophyll cell when covered by a bundle sheath cell, it is very good. So now we are coming to the C4 cycle, and both cell you will find there. So it's really good. So yes, advantages you can see. Mesophyll have the small chloroplast, so they are involved in the light reaction. Okay, and they have the so many enzyme which fix the carbon dioxide like carboxylation and bundle sheath cell. So cyclic phosphorylation take place here. So if cyclic phosphorylation take place here, so it means energy will also produce and no oxygen will be evolved here. So main thing is what? Main thing is it help in the increasing the carbon dioxide concentration. When the Rubisco, or you can simply say that bundle sheath cell and mesophyll cell both are very good and advantageous. And so having the Karan's anatomy, it is good for the plant. Having the Karan's anatomy. Okay. So now we
Mhm.
are coming to the C4 cycle. So C4 cycle, it is an alternative pathway present in the plant, okay, through which they can minimize the rate of photorespiration. I told you, photorespiration is a wasteful process. They are taking a different pathway. Photorespiration is not a good process. I will explain you how. It is going into the like many steps, and that step, and ultimately they are going to combine forming the those products which we already formed in the C3. So as of now, just remember, photo respiration is a bad process, so we have to minimize it. So C4 cycle is
Photo
minimizing the,
respiration is bad.
yes, sorry, yes. So photo is, we have to reduce the photo respiration. So C4 is reducing the photo respiration, so it is good or bad?
C4 is good because reducing bad.
Yes, it is reducing the photorespiration. Very good. So carbon dioxide first fixed in the mesophyll cell, and then it is released into the bundle sheath cell. It means in the C4 plant, not only mesophyll cells are there, but also the bundle sheath cell. Okay. So Rubisco is also very well protected from the O2. So Rubisco, always remember, you have to protect your Rubisco from the oxygen. See here,
Wait,
Rubisco
what
is
is
come.
Rubisco?
Yes, RUBP.
Ruth.
One second. RUBP is
Oh,
there. So RUBP when combining with the carbon dioxide CO2, Rubisco. RUBP combining with carboxyl carbon dioxide. So it is done by the Rubisco enzyme.
I see.
When RUBP is combining with CO2, this step is called
Mm-hmm.
carboxylation, and this is also done by the Rubisco enzyme. Rubisco, RUBP plus carbon dioxide, Rubisco.
Oh, I see.
Okay, so remember, Rubisco, for the Rubisco, you don't need to, you don't have to give oxygen, because whenever they see oxygen, they will become mad. They are not working. So that's why. So our ultimate aim is what? To protect the Rubisco from the oxygen,
Um,
right?
yeah, oxygen.
Because if they take the oxygen, then they will do the photo respiration. And photo respiration is,
Photo respiration is bad.
yes, that's why. So that's why we have to protect our Rubisco from the oxygen. Okay. So C4 cycle take place in how many cells?
6.
Now, how many cell? Two cells. First is
2 cells.
mesophyll cell,
And
another
then
is
there's a
bundle
bundle
sheath.
sheath.
Yes, but a C3 only take place in mesophyll cell. Okay,
Hmm.
but the cycle which you just saw, this cycle is taking place in the mesophyll cell only. Okay, carboxylation, reduction, and degeneration. But if I talk about the C4, it is taking place in both the cell. Okay, can you just see mesophyll cell and the bundle sheath cell, and they are well connected with the plasmodesmata. Like one cell has a connection with the other, and this is called the plasmodesmata. Can you just see that, Vishnu?
Yes, I can see it.
Okay, remember, atmospheric carbon dioxide is coming. Okay, so actually it this carbon dioxide forming the bicarbonate, and there is already it has the phosphoenol pyruvate, CCC. This is carbon number 3. I mean it contains the 3 carbon, and 1 carbon dioxide is coming. So just tell me, they are fixing each other. They are fixing both of them, carbon dioxide and 3 carbon compound phosphoenol pyruvate. This is called the PEP, phosphoenol pyruvate, PEP, PEP. So PEP is combining with CO2. So just tell me how many carbon will get formed after combining, after fixation?
Can you reiterate that? You cut out.
What?
Your voice cut out like
Oh, okay, okay, okay,
internet.
okay, okay. This is PEP, phosphoenol pyruvate, and this phosphoenol pyruvate is made up of 3 carbon. You
Okay.
remember glucose converted into pyruvate, so it is always 3.
Mm-hmm.
So it is a 3 carbon, and this is the carbon dioxide coming from atmosphere. So this
Mm-hmm.
3 carbon and 1 carbon is combining. So how many
Four.
carbon will get formed? 4 carbon. That's why it is called C4
C4
cell.
cycle.
Why? Because the first product is of,
Four
yeah,
carbons.
yes. So
Just
first
like
product,
C3 is like
yes,
yeah.
very good, very good. Just like C3, the first compound was C3, it is made up of 3 carbon. Same in the C4, the first compound is C4. And this is C4 compound is known as oxalic acid. They haven't right here, but you can just remember oxalic acid. Please say it, oxalic acid.
Oxalic acid.
Yes. Now it is transporting to the bundle sheath cell. Okay. Now it is coming to the bundle sheath cell. Now when it is coming in the bundle sheath cell, so it's a decarboxylation. It means what? D carboxylation, decarboxylation. It means we are
Take back
removing
the
again.
carboxylics.
Yes.
Yeah.
So from the C3, one carbon dioxide is going again in the Kelvin. It means inside the C4, C3 is also there, kind of, because this carbon dioxide can enter into the Kelvin, which is the C3 cycle. So one carbon dioxide is released. So now it becomes the C3. It by one carbon dioxide is taken out, na? So it becomes C3.
So 4 - 1 is 3. Yeah.
And now it is transporting again, and it becomes the phosphoenol pyruvate, 3. And again it is combining with C4, it becomes C4. It remain continue. So it means it is also seeing fixation, transportation, and regeneration. Okay, fixation, transportation, like from this to this and from this to this, transportation, and then regeneration. Okay, and decarboxylation also take place. So it means why we are doing this. We are doing this because in the Kelvin cycle, they are producing glucose. So C4 cycle has a very complex phenomena. They are producing CO2, and the CO2 is forming the glucose here, like when
Yeah.
they enter the Kelvin. So just tell me, C3 have the C4 or C4 can have the C3?
C4 can have the C3.
Yes, because glucose form will take place here, and
Both.
and yes, and which require the two cells,
Say it again.
which require the two cell, C3 or
This
C4?
one.
C4.
This
Okay,
one. The one.
so
Yeah.
C4 is very very important. It consumes some more energy per molecule. This is the very efficient because it has the Karan's anatomy. I told you Karan's anatomy is very good arrangement of cell, and this
Yeah.
is better adapted for the high temperature. Like C4
Hmm.
cycle is very very
High
much adapted.
intensity, right? High
Yes,
intensity too.
even it can work at the low concentration of carbon dioxide. It can work in very high temperature condition. It means it is not getting damaged. It is completely working. Very tolerant. It is very strong. So C4 cycle is very better, very much, you can say much better than the C3. Okay, so C3 it occurs at the normal temperature. C4 it occurs at high temperature. Does it have a Karen's anatomy? That arrangement, special type of arrangement? This one. You saw the arrangement. This is very good for the
Transition
photosynthesis.
is good. It's not there.
So Karen's anatomy present in the C3 or C4?
C4.
C4. So it leaves have the Karen's anatomy, very special arrangement, but C3 doesn't have. Only one type of chloroplast, but they have the two type of chloroplast. Remember, the first thing is RUBP, right? Which combined with the CO2. You remember?
Yeah.
But here
Ribulose.
Rubisco. Okay, but here it is PEP, the first acceptor which is combining with the carbon dioxide. So in the C3 it is RUBP, ribulose bisphosphate, and here it is phosphoenol pyruvate. You see na, phosphoenol pyruvate, PEP. I told you. I told you. Okay.
Yeah, you did. You did.
Okay. So the first carbon dioxide acceptor is PEP, and in the C3 it is RUBP. RUBP, ribulose 1,5 phosphate. So that's why. And why it is called C3 pathway
Oh, because the first product of the reaction is 3 carbons.
and C4?
Same thing, but for four.
Yes. Okay. So this is all about the C3. So remember, you have to learn the reaction, okay, like the transportation, and because in the examination they can ask you anything, like what is happening, plasmidesmata, mesophyll, bundle sheath. So it is all about the C3 and C4 cycle.